Oops. Not what I wanted to. Sorry about that. There you go. So, let's do part 2 of this. So, now that we're done with innate, we're going to talk about adaptive. And adaptive immunity or defense is a specific defensive system that eliminates almost any pathogen or abnormal cell in the body. And so, what it does is it helps to amplify the inflammatory response. And it's also important to activate complement. You remember the complement protein that we talked about earlier. Now, it does have some shortcomings. The problem is it must be primed, which means it needs an initial exposure. And why that can be a problem is because it takes a lot of time to do that, right? And so that's the only bad thing about adaptive defense, which is the reason why we have both the adaptive defense and the innate defense happening together. The innate defense is basically the first front that comes to start activating against stuff, and the adaptive defense, if it hasn't seen it before, it's going to start working on doing different procedures so that in the future it can recognize it easier. So there are characteristics of adaptive immunity. The good thing about it is that it's specific. If we have to prime it against some kind of particular antigen or initial exposure to a foreign substance, more specific, that it's going to end up being specific. So it's going to recognize, essentially target, very specific antigens. Okay? It's systemic. It's whole body, right? It's not restricted by the first initial site. And it has memory. It means that it can mount a stronger attack against something that's known, right? So this, and a known antigen is what it is, right? And so the second or subsequent exposures to it, it's going to be able to attack it very strongly. And so there are two main branches of this. There are both the humoral or antibody-mediated immunity and the cellular, which is cell-mediated immunity. So let's talk about the humoral immunity really quick. So this has to do with antibodies. The antibodies are produced by lymphocytes and they circulate freely in the body fluid. Right? They bind temporarily to target cells. So they're temporarily inactive. When they end up binding temporarily to target cells, making the cells inactive, and they mark them to be destroyed. And if you remember, how do we destroy them? Well, we can eat them with the phagocytes, right? Or we can go through complement, which is, labeling them and then putting the max within the cells to essentially lyse them. The humoral immunity has extracellular targets. Now, the cellular immunity or cell-mediated immunity, this has lymphocytes. Lymphocytes actually act against target cells, and so they can act directly. And if they act directly, they're basically killing the infected cells, or they can act indirectly. And basically, when they act indirectly, they're releasing chemicals. And those chemicals will help to enhance or increase the inflammatory response. Or they can act indirectly by activating other lymphocytes or macrophages. And here, the cellular immunity actually has cellular targets. Now, the targets that we're talking about with the humoral on basically and what they create or what they're targeting are antigens. And these are substances that can mobilize adaptive defenses and provoke an immune response. And so they're basically targets of all adapted immune responses that we have. Most are large, complex molecule, but normally found, not normally found in the body or the non-self, right? And the characteristics of antigens are basically they can be complete, antigens, or haptens, which are incomplete. They can contain antigenic determinants and can be self-antigens. So we're going to talk about each one of these in terms of what they are and how they basically are involved in this response or adaptive response. So complete antigens and haptens, what are they? Well, complete antigens have two important functional properties. They are basically immunogenic, which means that they have the ability to stimulate proliferation of specific lymphocytes, right? So they help with lymphocyte proliferation, or making new lymphocytes, right? And they're also, they have properties of reactivity. And so they're able to react with activated lymphocytes and antibodies released by immunogenic reactions. And so they react with activated lymphocytes / antibodies. So these guys right here, examples that you can see are like foreign proteins. You can think about any kind of lipids, those fats, or DNA, nucleic acids from other cells, or carbohydrates, polysaccharides that you can see on the membranes of different basically microorganisms. Or you can even see like pollens or microbes themselves. So those are all complete antigens. Incomplete antigens, basically these involve molecules that are too small to be actually seen. So they're not immunogenic by themselves. To become immunogenic, they have to attach to the body's own proteins. And so when you get protein plus hapten, you get UG, right? And so these are just some examples, or like hormones, or very small peptides, or even nucleotides. Now, they're going to cause an allergic reaction, and this is an immune system to mount an attack that is harmful to a person because it attacks self-proteins as well as haptens. So that's the problem with incomplete antigens, is that they can combine to essentially cause this huge immune response that can be harmful to you. So if in incomplete antigens, if they target or attach to a protein that your body naturally produces, and then this small molecule attaches to it and activates the body's sense to see it as foreign, then it's going to start attacking self-proteins. And if it's attacking self-proteins, it's basically your body is attacking yourself, right? So example of this that can happen is poison ivy, animal dander, detergents, or even some cosmetics can come in, and the small molecules from these things attach to a molecule that's already in your body, and that's why, and then your body sees that combination as it being bad and starts attacking it, and so that's why you get these crazy allergic responses. Now, they do also have within this is the characteristics of antigens is that they have this antigenic determinant. And so what that is that it's a part of the actual antigen that the antibody or the lymphocyte receptors bind to. So most naturally occurring antigens have numerous antigenic determinants. So these are the little determinants. These are the antigen binding sites, right? From the antibodies. So here is antibody here. This is the antigen, so it recognizes this, right? And these are the basically the antigenic determinants. So these are parts of antigens that antibodies or lymphocytes receptors bind to. They help to, basically they can mobilize different lymphocytes and then they can also help to form different kind of antibodies against them. And so they're large, they're chemically simple molecules that have little or known immunogenicity to them, but they help to determine these kind of foreigners. And so when we're talking about making antibodies and basically having antigen presenting cells, to do this, the adaptive immune system involves three crucial types of cells. There's two types of lymphocytes, both B lymphocytes and T lymphocytes. For short, they're known as B cells and T cells. B cells are known for the humoral immunity, and T cells are known for the cellular immunity. So B cells are there to help make antibodies, and T cells are there to help attack and get rid of the microbes. There's also the antigen presenting cells. They do not respond to specific antigens, and they play kind of these auxiliary roles in immunity. And so when we talk about lymphocytes, we want to talk about how do we get lymphocytes? Where do they come from? How do they become mature and able to fight? And then once they're ready to fight, they're just sitting there. How do we activate them? And so T and B lymphocytes share common development and steps in their life cycle. And there's 5 general steps. There's an origin, the maturation, the seeding, encountering and activation, and then proliferation and differentiation. And so let's actually look at this and what's going on. So if we look here, guys, the origin, where the heck do they come from? Well, we've already talked about this. Both B and T lymphocyte precursors originate in the red bone marrow. And so they're basically come to existence there, but then where they actually mature are in 2 different sites. If you notice over here, you have the lymphocytes, and then one type is going to come over to the thymus and maturate there. And so the lymphocytes precursors destined to become T cells migrate in the blood to the thymus, and then the B cells mature in the bone marrow. So I remember T for thymus, B for bone marrow, for where you know. So during maturation, lymphocytes develop immunocompetency and self-tolerance. So immunocompetency is the idea that lymphocytes can recognize only one specific antigen, right? So B and T cells display only one unique type of antigen receptor on the surface when mature and combine only one specific antigen. So they don't bind a whole lot of them. And they become what's known as self-tolerant. So they have to be unresponsive to their own antigen. Guys, they have proteins on the outside of their cell as well. And so you want them to not respond to their own antigen or they're going to self-destroy. So they become self-tolerant. They're like, you know what? I'm okay with myself. I'm not going to kill myself. It's basically what makes us successful as individuals, right? We try to say we have overall one job, and we go to like every single day I come. And this is kind of a silly example and not always true, right? But I've one job here at Lone Star. I go and, I have one office, and that's kind of my immunocompetency is the idea that I'm a faculty member. That's what I do. I'm not a faculty member, and I'm not a dean, and I'm not a VP, or I'm not a president altogether. I am one. So that's the idea that they are immunocompetent and they have a job, and their job is to that one antigen or that one specific area that they can bind. And then they're self-tolerant. Me, I'm okay with myself. I don't self-loathe myself, so I'm going to keep around, and I'm not going to, self-injure myself or hurt myself. And so that's the same idea with self-tolerance. They do have antigens on them, but they're unresponsive to their own. And so T cells here actually mature in this thymus, and they have both positive and negative selection processes. this up here. Right? So their positive selection process that they have as they mature is basically that they are capable of recognizing these what are called self-MHC proteins. If they can't recognize them, they'll be destroyed, or they'll destroy themselves by apoptosis. They also have this negative selection process as they mature. And so they basically will, if they bind to their self antigen, they'll go into apoptosis. This is a process here called clonal deletion. And so this is what ensures self-tolerance. So positive selection, they're like, okay, you know, we want to make sure that you have this particular protein on you that says that you are a T cell. If you don't have this, you can't pass, you can't go ahead and kill yourself. But we also want to make sure that you're not going to later go cuckoo and then end up killing yourself or other T cells. So we want to make sure you don't self-bind your own antigen. If you do, we're going to get rid of you. And this is our process of calling, if you see it looks like a colony, right? Clonal deletion, because we want to make sure that you're a good soldier that isn't going to cause problem later. And that's going to be what ends up telling us that there is this self-tolerance. Okay. And so at this point, T cells here, B cells here, we're mature. And so then we're going to go, and just to let you know, we know a lot about T cell maturation. We don't know a whole lot about B cell maturation. Only aminocompetent B cells are allowed to mature. Those that are self-reacted are eliminated by apoptosis as well, called the clonal deletion, but we really don't know how they do it. In the T cells we know what kind of proteins are there that they interact with. Now the seeding or the secondary secondary lymph node organs in circulation. So once this happens, right, the aminocompetent but still naive lymphocytes leave the thymus and leave basically the bone marrow, and they seed the secondary lymph node organs circulating through the blood in the lymph nodes, or the lymph, right? So they're called naive because they haven't been not exposed to antigens yet. They've never been in a fight. They've never been in a war. They don't know what their enemy looks like. They're naive. And so they're moved from these primary lymph node organs, right, the bone marrow and the thymus, and they're seeded to these secondary. Remember, we talked about that in the last unit. And so the lymph nodes in the spleen, right? And so this is going to increase the chance or the encounter. So The next thing we're going to have is antigen encounter and activation. And this is when the lymph nodes or the lymphocytes, the antigen receptors actually bind the antigen. And once that happens, the lymphocytes can be activated. So if the correct signals are present, if the antigen binds and everything is working right, the lymphocytes will complete its differentiation into active cells. And then the process of proliferation and differentiation happens. And so this is when activated lymphocytes proliferate, that's multiply, replicate, right? And then differentiate into effector cells and memory cells. Memory cells and effector T cells circulate continuously in the blood and the lymph and throughout the secondary lymph organisms. So effector cells fight. Memory cells help to basically respond later to the same antigen. So it's going to be way more quick the next time it encounters it. So B and T memory cells and effector T cells are constantly circulating. So, antigen receptor diversity. Basically, what determines what foreign substances the immune system will recognize? And a simple word for that is genes. Genes, not antigens, determine what foreign substances the immune system will recognize. Variety of immune cell receptors are a result of acquired genetic knowledge or microbes. There are 2,500 different gene codes for up to 1 billion different types of lymphocytes antigen receptors. So think about that. 25,000 different genes code for up to 1 billion different types of lymphocyte antigen receptors. There is a huge variety of receptors. Gene segments are shuffled around, resulting in a lot of different combinations. So let's do an overall review and look overall of B and T lymphocytes. And so you can see the types of response for B. You can see hormonal, since that's the antibodies, and you can think antibodies, B, and the T is going to fight the cells, right? Antibody secretion happens in B cells, not in T cells. Primary target for the B cells are extracellular pathogens. This is bacteria, fungi, parasite, and some viruses. And basically for T lymphocytes, intracellular pathogens are virus and infected cells and cancer cells. Sites of origin is the same for both, but the B matures in the red bone marrow, where the T matures in the thymus. The effector cells, the fighter cells, are going to be basically for the B lymphocytes are these cells called plasma cells. And the effector cells, there's more. There's cytotoxic T cells, helper T cells, and regulatory T cells that are all part of the fighters or effector cells in the T lymphocytes. Both do have memory cell formation. So, antigen-presented cells, the APCs, these are going to be there to engulf antigens and present fragments of antigens to T cells for recognition. Right? So they're going to help basically get rid of some of them and then go, hey, look, you need to go fight this dude, and this is what he looks like. There are major types: dendritic, macrophages, and B cells. So let's talk a little bit about dendritic cells. They're found in connective tissue and the epidermis. They act as mobile guides of boundary tissues. And how do they work? Well, they work by phagocyzing pathogens. They phagocyze the pathogens that enter tissue. And then enter the lymphatics to present antigens to the T cells, right? This is the most effective antigen presenter. He is, if you could have any antigen presenter in your world, it would be a dendritic cell. And this is a guy is the key link between innate and adapted immunity. Now, macrophages, we've talked a lot about those. They're widely distributed in connective tissue and lymph node organs. They present antigens to T cells as well, which not only activates T cells, but also further activates macrophages. Activated macrophages become ferocious. This is my favorite part of this. Phagocytic killers. Right? They also help to activate an inflammatory response. And it's very powerful, right? But these guys, all of these guys are terrifying. You would not want to meet them if you were a bacteria walking down the street. You would not want to encounter any of these because you're dead. I mean, it's awful. It's kind of like the Keanu Reeves movie where the mob accidentally kills his dog. And then he goes and kills everybody in the movie. I'm sorry if you haven't seen it by now. It's too late. It's old. But there are people that, these are cells you would not want to come in contact with. Now, so now let's talk about B lymphocytes and T lymphocytes specifically, since we talked about these kind of helper cells that are there. So B lymphocytes, how are they different than others? Well, we just got done talking about all the dendritic cells and the macrophages. What do? They help to activate naive T cells, right? Naive T cells haven't seen any outsiders yet. So basically the dendritic cells and macrophages, they go out. It's like if I wanted to make you a hitman and I wanted you to see, okay, there are certain, for instance, a stormtrooper. I'm in the Star Wars universe now, right? I could cut the head off a stormtrooper, show you that, and say, now go kill these people. And then at that point, you know the stormtrooper is bad, so I need to go get rid of those. And you know what it looks like, so you go out and get rid of it. That's what everything else has been doing. But these B lymphocytes don't activate T cells. So they're a lot different, right? What they do is they present antigens to T cells to help end their own activation. I know that sounds really weird. Just hang with me here. When B cells encounter target antigens, it provokes what's known, and we talked about this for a second, a humoral immune response, aka antibodies. So we make antibodies for the antigen presented, right? The B cell are activated when the antigen binds to the surface receptor. That cross-links them. And so most of these clone cells that we get from them end up becoming plasma cells. And the plasma cells end up making antibody for the body. So let's look at this really quick. So if you see here, right, let's talk about getting into adaptive immunity, going into humoral immunity. So if you look here, the primary response, this is the initial encounter with the antigen. Here is the antigen that you can see on these guys here, right? And so this antigen is going to bind to a receptor on a specific B lymphocytes. If you have a B cell here that doesn't have the complementary receptor, it won't bind, right? They're free. So once that happens, they bind, what's going to happen is they're going to perform or start proliferating to form colonies of these guys. Because if there's antigens present, then we might need more cells to help with the attack. And so then what they're going to do is they are going to go and create plasma cells. And these are effector B cells. And basically what these are going to do is they are going to secrete the antibodies so that the antibodies can then determine or help with recognizing these antigens or these foreigners. Now, one of these will be a memory cell. And those will help to prime for a response to the same antigen later. And so then we can go and look at the memory cells. What happens there? And so you can see we have this memory cell here. And we're going to have clones. And so basically what happens is if this memory cell, and so this is a secondary response. This is later. What happened earlier was a primary response. The very first time you ever come in contact with that antigen or that foreigner, right? This is what happens later. What if that same foreigner comes back into your place, right? And because you have this antibody, you have a picture of what it looks like. So you can respond to it faster. And so basically what happens is that antibody binds. It can start cloning cells immediately to make the ancestral cells that we used to have. And so you can see now, instead of having a few and it took forever, we have all these plasma cells secreting antibodies to basically warn everybody, this guy is terrible. Get him out of here. Here's the picture. Here, go, 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 go. And then we have additional memory cells because we know, okay, this could be a problem in the future. Now we've seen it a second time. Let the secondary response be quicker from here on out, just like it was this time. So that primary immune response, that basically the proliferation or the replication of those cells and the differentiation upon exposure to the antigen for the first time, it has a lag period about 3 to 6 days. And the peak level of antibodies is reached about day 10. And then after that, the antibody levels decline. And so when we get to the secondary response, this is re-exposure, right, to the same antigen. And so it's going to be faster. It's going to be more prolonged, and it's going to have a more effective response. And so the sensitized memory cells, the ones that we have, provide what's called immunological memory. This is going to respond within hours. It's not going to take the 3 to 6 days. Antibody peaks, actually, the antibody levels peak in 2 to 3 days at a much higher level than they were the first time, not 10. The antibodies bind with a greater affinity. That means they attach to it and hold on to it better. And antibody levels can remain higher for weeks to months. So if you can see here, right? This is looking at a graph, and here it's talking about how many antibodies are there, the concentrations that you see. And so basically here you have for your first exposure to A. And so you can see about day 6, you'll start getting antibodies peaking at 10, right? And then you can see it go down. And then at day 28, you're exposed to that same antigen again. You can see, boom, within a couple, 2 to 3 days, you're getting this huge amount of antibodies. And look how high it is. Now, something that it hasn't seen before, it's going to react the same as it did the first time. Do you see how these are the same? So these are a good graph on looking at secondary and primary humoral responses. And we are getting close here. So let's talk about active humoral immunity. This occurs when B cells encounter antigens and produce specific antibodies against them. And there's 2 types. There's naturally acquiring and artificially acquiring. And so naturally occurring means that you've encountered it. You've actually formed in a response to an actual bacterial or viral infection. Right? Artificially acquired is due to a vaccine. You weren't naturally around it, right? And vaccines are basically dead or attenuated, which means they're not as lethal. or as high concentration, or able to replicate as well. They've done something to it to make it less effective. And so when they do this, basically what they're doing is giving you or providing antigenic determinants, right? That are immunogenic and reactive. And so this part spares us from the primary response. So that's naturally acquired immunity and artificial acquired immunity. Now we have what's called passive humoral immunity, and this occurs when ready-made antibodies are introduced to our body. B cells are not challenged by antigens. Immunologic memory does not occur. Protection ends when the antibody degrades. And so there's two types of this. And so passive, think about this. The antibodies for naturally occurring antibodies are delivered to a fetus via the placenta or in the infant through milk. So this is the mom providing them, right? And the artificial is some kind of injection of serum. And so they may try to inject you with GABA globins. And so this protects you. Until they degrade. So you're hoping, breastfeeding helps a lot because you're getting the immunity. So what's happening is those antibodies are there. So your baby, when they're exposed to some kind of foreign entity coming in there, right? You have your immunity that's going to help protect it. And in the process, it's going to be able to make their own to basically combat those. But they're not going to get as sick because they have yours to work with for now. And so here's a humoral immunity. You have both active and passive. If you can see, active is basically being exposed to it in some way. Naturally acquired means that you've actually come in contact with it in the world. Artificial means that you've been giving some kind of vaccine or been vaccinated. Passive means that basically you're not acquiring it from being exposed to it in some way. So basically you're naturally acquiring through antibodies passed from your mother, or you are artificially acquiring it through injections. This one ends up degrading. And so it's for short term. So antibodies are also known as immunoglobulins. And so we've been talking about how our body makes antibodies, And it's like, what are they? Well, they're proteins secreted by the plasma cells. And they're capable of binding specifically with antigens detected by B cells. They are grouped into 5 different Ig classes, and Ig coming from immunoglobulins. And so this is what an antibody kind of looks like. You can see the proteins there. And there's 4 chains. And so they consist of, basically, you can see heavy chain here, light chain here, light chain here. And then you can see on here, there is a variable region. And the variable region here is that antibody or antigen binding site. Okay. And so on here too, you'll also notice this last part is this basically what's known as a, so you have the variable regions, and let me go. And then you have the constant regions. And so, go on here. Let's see better. And so if you look on the kind of dark spots, those are going to be the variable. And so the variable regions are the regions that are the antibody, the antigen binding areas. Now these parts that are lighter that go through here on both sides, right? Those are what are known as constant regions. This region is what ends up determining the antibody class. They are basically considered effector regions, which means they serve the common functions of all antibodies. And basically they do it by dictating the type of cells or chemicals the antibody binds, and how the class functions to eliminate the antigen. And so these antibodies are all over. Some fix complement, some circulate in the blood, some are found in body secretions. It all depends on the type of antibody. And so this kind of gives you an idea of what the structure looks like a little bit more. And so depending on the class, like IgMs or the first antibodies released, they're potent agglutinating agents. And you remember agglutination when we talked about blood types, and what happens when the wrong blood gets in and it kind of falls out. So that kind of explains that. And they readily fix and activate complement. IgAs, these are secretory IgAs, and they're found in the mucus and other secretions. They help prevent entry of pathogens. IgDs, these are functions as B cell receptors, right? IgGs, they have 75 to 85% of the antibodies that are actually in the plasma. They're from that secondary and late primary response, and they can cross the placental barrier. And IgEs basically are active in allergic and parasitic allergies or allergic response and parasitic infections, and can cause mast cells and basophils to release histamine. And basically, from here, I just kind of if you look, I just wanted you to see, you don't have to identify what they look like, but just to know that not all antibodies look exactly the same. I showed you the general functions, and you can see what I showed you earlier. There's a bunch of them on this guy, right? And so they all have that basic shape to them, but they're put together slightly differently. Now, the plasma B cells can switch from making one class of antibody to another. So basically, certain one, you have antibodies like IgM is released during primary response, but plasma cells can switch to IgG for a secondary response. So they retain the specificity for the same antigen, but they can switch the type of antibody that they're making depending on kind of what needs to happen. Almost all secondary responses are IgGs. And just basically switching can occur. So, antibodies basically do not destroy antigens. They just inactivate them and tag them so that they can be targeted to be degraded. So the defensive mechanisms used by antibodies are neutralization, agglutination, precipitation, and complement fixation. So let's look at those and see what I mean by each one of those. Okay. So when you make this antigen-antibody complex, so you have the antibody here. Why is this not working? Sorry, guys. It's getting out. It's coming loose. When you have this antibody come here together with this antigen, right? Things can happen. So the first thing this antibody-antigen complex can do is it can fix complement fixation and activation. And so this is the main antibody defense against cellular antigens, right? So basically, on here, you talk about bacteria or mismatched red blood cells. When several antibodies are bound close together on the same antigen, complement binding sites on their stem regions are aligned. This alignment triggers complement fixation, which, if you can see, we got a MAC there, right? And then that will lead to that basically pore allowing water to come in, and then basically cell lysing, right? It can also lead to these other phagocytosis, right? Or this increase in inflammation. And so, also what can happen is these other things. And so this antigen-antibody complex can inactivates by neutralization. So this is basically trying to get rid of it, figure out what it needs to do. Here in neutralization, it's the simplest but the most important defensive mechanism. And basically the antibodies block specific sites on the viruses or the bacterial exotoxins and prevent the antigens from binding to receptors on tissue cells. And so that means they're not going to be able to get into those cells. Agglutination may happen too. And this is when antibodies combine the same determinant on two or more different antigens at the same time. Do you see how they have all these red blood cells there, right? On here, each antibody has two arms, each containing a variable region capable of binding one antigen. This allows for the antigen-antibody complexes to become cross-linked into these larger lattice-like clumps. And this basically is agglutination. So this is, remember, all the clumping we get from mismatched blood. And the last possible activity that can happen is precipitation. So this is when you get soluble molecules instead of cells become cross-linked. They fall out of solution. This makes it easier so phagocytes can work. And so all of these are working together to at least try to help phagocytosis, or, the destruction of these foreign entities. Inflammation, chemotaxis, where in histamine release, let's get the neutrophils, let's get these different cells there so that we can tag them. So then we can have basically first neutrophils there to get rid of them. Of course, those act as phagocytes as well, and the macrophages there. And then let's get the T cells and all the helpers to activate so we can get rid of those things. And then last but not least, let's lyse the cell. And so T cells provide defense against intracellular antigens. Some T cells directly kill cells, others release chemicals that help regulate immune responses. T cells are more complicated than B cells, both in their classification and their function. And so we can separate T cells into two populations based on their glycoprotein receptors that are on their surface. And so these guys, did I talk about them? Yeah. So let's talk about the major types of T cells. So you have this immature lymphocyte that starts in the red bone marrow, and then they mature, and you can see in the thymus they're going to either be CD4 or CD8 cells. And so when a CD4 cell basically matures, they become activated by these dendritic cells, the APC cells. Remember, we talked about how they can come in and they're the helper cells. And then from there, they divide. And so these CD4 cells usually become helper T cells. And the helper T cells help to activate B cells. Also, other T cells and macrophages. That's a direct adaptive immune response. Some become regulatory cells. And so they help to moderate the immune response. But they can also become memory cells, right, to help with secondary exposure. Now, on the other side, you have a slightly different class of protein that's on these dendritic cells that come to activate. If you notice, this is a class 2, this is a class 1. And so the dendritic cell comes in, helps to activate them. So most of the time, CD8 cells become cytotoxic cells. And these guys are capable of destroying cells that harbor foreign antigens. Again, they can also become memory cells. the helper cell, the regulatory cells, and the cytotoxic cells are all activated T cells. The naive cells that we were talking about that haven't been exposed yet are termed CD4 and CD8. Remember how we were talking about that at the beginning? So if we look at the helper T cells in the humoral response and the helper T cells with the cellular immunity, you can see here, right, what they do. Basically, these helper T cells bind with the self-nonsense complex as a B cell that has encountered its antigen, and it is displayed on this secondary surface. And so in this case, the only thing that we're looking at here that's most important, I really want to talk about this. In here, we're just noticing, I don't want to get too in detailed here with you guys, is that this helper T cell is basically helping to activate this B cell by allowing it to essentially interact with its displaying antigen. And basically what happens here with the helper T cell with cellular immunity is you have these helper T cells here. They bind to this dendritic cell. This dendritic cell is going to release molecules, those interleukins that we talked about earlier. That's going to allow the co-stimulatory molecules to be expressed. And then essentially that's going to help activate the CD8 cells to become the particular helper T cells in cellular immunity. And so let's watch this quick film to kind of talk about immunology in general. Oops, nope. Why isn't it working?
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses, ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.
And you can look through here, and this is just a summary of everything that we just talked about. And so let's go through the last bit of this and talk about all these homeostatic imbalances. And so pus, what is pus? I'm going to do this. Basically, pus is just that yellow mixture of dead neutrophils, tissues, and cells, and either living or dead pathogens. An abscess is just basically when collagen fibers are laid down, walling off sacs of pus. And these may need to be surgically removed. And so abscesses can be a problem because things like certain types of bacteria, like tuberculosis bacilli, are resistant to digestion by macrophages and remain alive inside of them. So you have to be careful. Now, you can get these things called granulomas, which are tumor-like growths, right? And basically, these are areas of infected kind of macrophages that are surrounded by uninfected macrophages in an outside capsule. So in these, bacteria may remain inactive forever, or if a person's immunity decreases, may break free and become activated and cause disease again. Now, immunodeficiency can be a congenital or acquired condition. And basically what happens here is an impairment of function or production of immune cells. So, severe combined immunodeficiency syndrome, or SCID, is under this, and it's basically a genetic defect. And it's marked by a deficit in B and T cells. Hodgkin's disease is an acquired immunodeficiency. That basically causes cancer. And it's of the B cells. And so basically, this is going to depress lymph node cells and thus lead to immunodeficiency. AIDS, acquired immunodeficiency syndrome, is caused by HIV or human immunodeficiency virus. And basically, it interferes with the activity of helper cells. So you can get severe weight loss, night sweats, swollen lymph nodes. And then you can have other types of immune disease. For instance, rheumatoid arthritis is an autoimmune disease. It's known to cause problems in joints. You have type 1 diabetes. Basically, type 1 is when you don't make insulin, and it's basically an issue with not being able to make the right receptors. And also you have multiple sclerosis, and multiple sclerosis is immune deficiency that happens due to the fact that your body starts seeing myelination, or the protection of the nerves, is bad, and it starts attacking that. And when you demyelinate nerves, you're going to decrease how fast that a action potential goes down, and so it's going to cause issues with nerve cell communications. And so we are at the end of our lecture for the immune response. Thank you, and I can find it. We will be doing the next unit soon. Bye.